Up next, we have a double header, literally. Here in the auditorium, we've got Geoff Dermott from 10x Genomics. I'll keep this introduction brief.
Oh, you don't have to introduce it all, that's fine.
Our next speaker needs no introduction, and off I go. Online, we have Ulrike Schumann from ANU, and please take it away.
Thanks, Tony. All right, hello. It's an absolute pleasure to be here today. So at 10x Genomics, we have two different but complementary spatial platforms. Both of these technologies are compatible with either fresh frozen or FFPE tissues, and at the highest level, both provide gene and protein expression data whilst preserving the spatial location. Since I've only got 10 minutes, I won't have time to compare and contrast these platforms, but instead I'm going to cover some new spatial product releases that we have coming for 2022. And I want to begin by discussing what's new to the Xenium platform. So just as a really quick refresher, here's an example of some data generated using Xenium. We're looking at an FFPE section of mouse kidney tissue. You obviously get the H&E image of the tissue, so you can still do all your visual annotation. But the real power of the platform comes from capturing the underlying molecular information, and of course, that's the spatially organized whole transcriptome gene expression from the entire tissue. We have 4 major Xenium product releases scheduled for 2022, as listed here. So we'll take a quick dive into each one separately over the next few minutes, but I'll definitely skip the larger capture area slides because I think the name of this product is pretty self-explanatory. If your tissue doesn't fit inside the current 6.5 by 6.5 millimeter capture area, then we have 11 by 11 millimeter capture area slides coming very soon. So we begin with our upcoming Xenium gene and protein expression release. Now you'll have the ability to capture whole transcriptome gene expression and protein expression from the exact same tissue slice. We'll accomplish this using panels of antibodies that are tagged with oligonucleotide barcodes. So for those of you who are used to doing sort of site-seq with single cell sequencing, it's a very conceptually similar approach. This approach basically converts a protein signal into a DNA signal, which of course means we can now use our sequencer to detect proteins. So we're looking at an example of protein and RNA detection from the same tissue. Here we have results from an FFPE breast ductal carcinoma tissue section. We used a multiplexed oncology panel of antibodies alongside whole transcriptome gene expression. As you can see, several of the new marker genes are presented here. The left-hand panel for each gene shows the spatial location of the corresponding mRNA transcript, and the right-hand panel shows the spatial location of the corresponding protein. You can clearly see the differing expression levels between RNA and protein, demonstrating the importance of assessing both modalities. And I think that CD20 over here on the top right is the clearest example of that. You can see almost no signal at the transcript level, but a very clear spatial profile at the protein level. And to reiterate this point one last time, with this assay, you're now getting whole transcriptome gene and protein expression from the exact same tissue slice. Another development we've been working on is called the CytAssist instrument. In the current Xenium workflow, you section your tissue directly onto the Xenium slide. So that means you pretty much have to commit to the tissue section before you actually get time to go and analyze it under the microscope. But the CytAssist will transfer analytes from your FFPE tissue section already on a plain glass slide onto the Xenium capture area. So what this means is you can section directly onto a plain glass slide. You can go and look at those sections on the microscope, and then you can choose which section you want to carry forward into the Xenium workflow. And then really importantly, and alternatively, it also means you can access pre-cut FFPE slides that you might actually have archived somewhere in the lab. Here's how the instrument fits into the Xenium workflow. Basically, you take the glass slide with the section of interest and place it into the CytAssist instrument. And next you place a blank Xenium slide into the CytAssist instrument. And you align the tissue that's on that plain glass slide over the top of the capture area that's on the blank Xenium slide. And then within the instrument, molecules are transferred from that tissue on the plain glass slide down to the surface of the Xenium capture array. And once that's complete, you basically follow the standard Xenium workflow to complete your sequencing library. This is a really great example of how you can choose the exact position of a tissue section to be aligned with the Xenium capture area. Here's a large mouse embryo FFPE section that was placed onto a plain glass slide, and then again, that plain glass slide was placed into the CytAssist along with a blank Visium slide. And then using the CytAssist, we were precisely targeted the head and the torso of the embryo to match a Visium capture area. Here's the spatial transcriptomics results from this sample. As an example, we have some organ-specific genes that were detected in the whole transcriptome data, and you can clearly see that these genes are being expressed in the regions we'd expect to find them. So the key take-home message is that when using CytAssist, tissue preparation for Visium becomes much more flexible and simple. The final new product I wanted to cover is probably one of our most anticipated products to date, and it's called Visium HD. And this will take the resolution of Visium from its current size of 55 microns right down to just single-digit micron resolution. So this will enable spatial transcriptomic profiling across your whole tissue section at single-cell scale. To achieve this massive jump in resolution for Visium HD, a completely new slide architecture needed to be developed. While our R&D team is still very hard at work, we're able to provide you with a feel for what the scale of Visium HD will be. So here we're zooming into a mouse eye, and you can see how the resolution and expanded tissue coverage of Visium HD allows for precise identification of layers within the eye. So for Visium HD, you get the power of whole transcriptome gene expression across your entire tissue section, but again, it will be at single cell resolution. That concludes our new product introductions for Visium, but with the final few minutes, I wanted to cover a completely new spatial platform that we've been developing at 10x Genomics. We call it the Xenium platform, and it will enable in situ sequencing. And this, of course, means that you can now detect your RNA and proteins within the tissue itself with subcellular resolution. Just like the Visium platform, Xenium will be compatible with both fresh frozen and FFPE tissue. And one key differentiator for this platform from other in situ technologies will be the sample throughput. Every 2 days, you can run 2 full slides, with each slide containing a capture area of 12 by 24 millimeters, which gives you the flexibility to place multiple samples on the same slide. At launch, we'll have a 400 gene panel where 50 of those genes can be customizable. And then finally, just like all of our assays, we'll have a suite of robust analysis tools and software to accompany the instrument. I wanted to quickly touch on the assay biochemistry as it's another key differentiator of the Xenium compared to other in situ platforms. So we'll take a high-level walkthrough of the straightforward sample preparation process. So you section your tissue onto a slide and then you place the cassette on top. And next you permeabilize the tissue before adding your probes. And then a probe specific to each gene transcript is then hybridized and ligated together to create a circular construct. And this probe hybridization and ligation approach is unique to Xenium. Since only a fully ligated and circularized probe will generate a signal, this approach ensures high specificity, which is something that can be lacking in other in situ methods. Next, we enzymatically amplify this circular construct, so you can see that we have converted a single RNA molecule now into a small ball of amplified DNA. And then later, fluorescent oligos will bind that amplified barcode sequence to identify the transcript. So this will result in a bright signal that's easy to image and has a high signal-to-noise ratio. And this is particularly important when working with samples that have high autofluorescence, which is something that's typical with FFPE. Now we'll take a look at some cool data from the Xenium platform. The first panel here shows results from the fresh frozen mouse brain. This has become a pretty standard sample type to show for any in situ sequencing technology, as you've all seen today. But because of the robust assay biochemistry that I just covered on the last slide, with Xenium it's possible to analyze such a wider variety of tissues. You can see here we also have some data from fresh frozen mouse kidney and FFPE human pancreas, which is actually a very challenging sample type. And then other examples of challenging samples, we've successfully analysed FFPE human lung cancer, colorectal cancer, liver, and fresh frozen human brain. And we'll continue to add to that list as we keep developing this platform. Finally, let's take a sneak peek at some data from our R&D team. Here we're analysing an FFPE sample of human breast cancer. We don't have time to cover the interesting biology that Xenium revealed today, but to fully appreciate just how amazing the subcellular resolution is, we'll zoom right into a section of this tissue. Here in the right-hand panel, you can see the subcellular tumour microenvironment identified by the Xenium platform. And from the Xenium data, we're able to identify single macrophages, lymphocytes, and cancer-associated fibroblasts based on their expression of cell-specific markers. So identification of such immune cells would obviously be very clinically relevant for the development of immunotherapies in cancer research. So with that, I'll say once again, thank you so much for having me here today. If you want to learn more about either our Visium or Xenium spatial platforms and any of the associated new products that I just talked about, then please just come and talk to us. We're out the back there. We've got a little booth. And with that, I'll now hand it over to Ulrika to present some very cool Visium data that she's generated on the mouse eye.